Gypsy's microdeletion, because I'm tired of y'all's misinformation. Save this for later so you don't lose it. Let's read it together as a class. A 1q21.1 microdeletion is a very rare genetic condition in which a tiny piece is missing from one of the body's 46 chromosomes. The tiny missing bit raises the risk of learning, behavior, and mental health problems and physical abnormalities. Keep in mind that in her medical documentation, every single time they mark her behavioral health, they say that her behavior was perfect and there were no behavioral concerns. And every single time they say there were absolutely no learning concerns either. But there is a wide individual variation. Keep in mind that in her medical documentation, in the chromosome medical documentation, they state that it can be asymptomatic, meaning that there are absolutely no symptoms whatsoever. Individuals with a 1q21.1 microdeletion range from people with no symptoms to others with developmental delay and health problems. Keep in mind that in her medical records, it states that there were absolutely no developmental delays. There were no developmental delays marked in her medical records every single time they said developmental delays. They put none. Genes and chromosomes. Our bodies are made up of billions of cells containing tens of thousands of genes. Genes act like a set of instructions directing our growth and development and how our bodies work. Genes are carried on structures called chromosomes. There are usually 46 chromosomes, 23 inherited from our mother and 23 inherited from our father. So we have two sets of 23 chromosomes in pairs. Apart from two sex chromosomes, two X's for a girl and an X and a Y for a boy. And chromosomes are numbered 1 to 22, generally from largest to smallest. Chromosome 1 is the largest of the chromosomes. Each chromosome has a short arm on the left in the diagram at the bottom of page 3, that'll be in the next part, called P for petite, the French word for small, and a long arm called Q on the right. Looking at chromosome 1q, chromosomes can't be seen with the naked eye, but if they are stained and magnified under a microscope, each one is a distinctive pattern of light and dark bands. Looking at chromosomes in this way, it is possible to see the points where the chromosome has broken and what material is missing. If the missing piece is large enough, a missing piece visible under the microscope is called a deletion. In the diagram at the bottom of page 3, you can see the chromosome bands are numbered outwards from the point where the long arm meets the short arm. In a 1q21.1 microdeletion, the chromosome has broken in two places, in band Q21.1, leaving out the chromosome material between them. This particular deletion is so small that it can only be identified using molecular or DNA technology, in particular a technique using microarrays, array CGH. This shows gains and losses of tiny amounts of DNA throughout the genome, also called duplications and deletions, and can show whether particular genes are present or not. A deletion so small that it can only be identified in this way is called a microdeletion. Copy number variant. 1q21.1 microdeletions are found in the general population, albeit rarely, as well as in people referred for chromosome testing. However, they are more common in people referred for genetic testing. A 1q21.1 microdeletion is sometimes called a copy number variant. People usually have two copies of each part of any chromosome, including chromosome 1. But it's quite common to have one copy or three copies of tiny parts of any chromosome. The 1q21.1 band of chromosome 1 is an area where people in the general population do sometimes have one copy or three copies. Having just one copy or three copies appears to make no difference to development in some people. But since a lot of people with a copy number variant and 1q21.1 do have developmental problems, it seems that the deletion can make some people more vulnerable to difficulty. The information in this guide is drawn from what is known about around 60 people with a 1q21.1 microdeletion aged between birth and 31 years. There are broadly two sizes of 1q21.1 microdeletion. The first type is smaller. It's called distal because it's closer to the tip of the long arm of the chromosome than the larger second type of microdeletion. A class 1 deletion spans around 1 million base pairs, or one so-called megabase of DNA. Base pairs are the chemicals in DNA that form the ends of the rungs of its ladder-like structure. Chromosome 1 has around 247 megabase in total, and band 1q21.1 alone contains around 5.4 megabase. In a class 1 deletion, DNA is typically missing between around 146 megabase and 147.8 megabase, as you can see in the diagram above. The megabase numbers show a position on chromosome 1 between position 1 megabase, the tip of the short arm, and position 247.975 megabase, the tip of the long arm. We know that the missing section includes at least 9 known genes, and there may be more. The second type is a larger deletion of around 1.35 to 2 megabase, including 25 known genes. DNA is missing between around 145.4 megabase and 147.8 megabase, as you can see in the diagram above. This includes a region of the chromosome known to cause a syndrome known as TAR syndrome. This yellow box is what we're reading now. TAR syndrome. Thrombocytopenia, reduced platelets in the blood, leading to spontaneous bruising and lengthy bleeding. AR, absent radius, the outer shorter bone in the forearm is missing. And if you look at the diagram below, that's the diagram that they were referring to in part 1. May receive a laboratory report including a karyotype. A karyotype is a way of describing what the chromosome looks like. It is likely to read something like this. 46, the number of chromosomes in your child's cells. XY, the 2 sex chromosomes, XY for males, XX for females. DEL, a deletion, that is, material is missing. 1, the deletion is from chromosome 1. Q21.1, Q21.1, the chromosome has 2 breakpoints, both in band 1q21.1. The material between those 2 breakpoints is missing. Array CGH report. Most often, a deletion of 1q21 is detected using a molecular technique. The laboratory will send a report that will usually look like one of these. Y'all can read all those numbers for yourself. 46, the number of chromosomes in your child's cells. XY, the 2 sex chromosomes, XY for males, XX for females. DEL, a deletion or material is missing. 1, the deletion is from chromosome 1. Q21.1, Q21.1, the chromosome has broken in band 1q21.1. ARRCGH. The analysis used microarray technology, all those numbers, 3 different markers whose position in the 1q21.1 band is known have been shown to be missing. ARR. The analysis was by array, ARR, comparative genomic hybridization, CGH. 1q21.1. Chromosome involved is 1 and the position of the deletion is in the band Q21.1. All those next numbers, the base pairs between 145167814 and 146090213 have been shown to be missing. Take the first long number from the second and you get 922.399, 0.922 mega whatever, or 922 kb. This is the number of base pairs that are deleted. HG19, human genome build 19. The human genome is updated as more information is found. Each new version is called a build, and each build the base pair numbers may well change slightly. In January 2013, HQ19 is the newest build. Confusingly, HG19 is also sometimes called Genome Reference Consortium Human Genome 37, GRCH37. X1 means there is 1 copy, and these base pairs, not 2, 1 on each chromosome 1, as you would normally expect. Are there people with 1q21.1 microdeletion who have developed normally and have no health, learning, or behavior difficulties? Yes, there are. Save this for later so you don't lose it. The 1q21.1 microdeletion can be silent. Some of the parents of children with a 1q21 microdeletion have the same microdeletion but do not have any unusual features, delayed development, or behavior problems. Other parents who also have the deletion are extremely mildly affected. Some children with a 1q21.1 microdeletion also develop normally. The effects of some genetic variant ranges from being barely perceptible to being obvious and severe. In this sense, they are like infections such as the flu that can be mild or serious. The most likely feature: small head, which we know that Gypsy has the microcephaly, increased possibility of mild or moderate developmental delay. We know that in all of her medical records, every time they click the box that says developmental delay, they've said no. Every time they click the box that says learning disability, they say no. Slightly unusual facial features, increased possibility of behavioral or mental health problems. We know that throughout all of her medical files, they continuously say that she's cooperative and that she has absolutely no behavioral problems or mental health problems. It's not until 2013 in Missouri that we even start seeing that slightly pop up. Heart problem, loose joints, or double jointedness. Other features: seizures, disturbances in the lens of the eye, increased risk of other inborn anomalies. Most likely features: small head. Studies have shown that around 2/3 of people with this microdeletion have a small head or a head that is small compared to their overall size. This suggests that the microdeletion influences brain growth. Some people with an extra copy of this section of chromosome 1, a microduplication, have an unusually large head, and this also supports the idea that the microdeletion influences head growth. Researchers have identified a gene called HYD1N that is likely to be important in determining head growth. This is what Becca Scoops is referring to when she says that doctors said HYD1N say that it contributes to a possum-like features. Like, no it doesn't, girl. No it doesn't. It has to do with head size. That's it. There is also a possible link between head size and mental health problems, particularly schizophrenia. This does not mean that everyone with 1q21.1 microdeletion and a small head will develop schizophrenia. On the other hand, it does mean that healthcare professionals should be alert to early warning signs. Now, that is quoting an article from 2008, and it's important to remember that the title of that article has to do with this very specific microdeletion and its correlation to schizophrenia. That entire article is only about schizophrenia. Among unique members with the microdeletion, head size is variable. 3 out of 7 members have a large or relatively large head, 1 is average, and 3 small. Increased risk of developmental delay and learning difficulties. Some children with a 1q21.1 microdeletion develop at a normal rate and cope well academically without support. Others are slow to reach their developmental milestones, need extra help at school, and may need special schooling. Remember that on all of Gypsy's medical records, anytime it said developmental delay, they said none. Behavioral concerns, none. Learning supports needed or learning concerns, none. This means that a baby or child with the microdeletion should be monitored vigilantly for delay, and extra help and therapies should be sought and offered promptly. The microdeletion does not make it possible to predict how mild or severe the delay and the learning difficulties will be. The spectrum includes people who have no noticeable problems and a few people who are profoundly affected. In most cases, the delay is mild or moderate. That is quoting a Medford 2008 study. In most cases, the delay is mild or moderate. Among unique members, the range of developmental delay is very broad indeed, from none to profound. Babies and toddlers may be late to reach their developmental milestones. Sitting, crawling, walking, and fine motor control and toilet training may also be delayed. Her medical notes say that none of that was delayed. I don't know though, I wasn't there. Low muscle tone was found in around a quarter of children in one study, as well as among unique members. Unique's experience is that when seizures have been hard to control, a more severe degree of learning disability has become apparent. Slightly unusual facial features. Most children and adults with 1q21.1 microdeletion look like other members of their family. They may, however, have 1 or 2 slightly unusual facial features, such as an unusually prominent forehead, deep-set eyes, and a somewhat bulbous nose, none of which Gypsy has. These features do not usually make them stand out, but they may mean that your child has a passing resemblance to others with the microdeletion. Unique families have commented that the broad forehead can make their child's face look triangular. Where more than one child in a family has the microdeletion, the unusual facial features are not necessarily the same. A gallery of adults and children with the microdeletion has been published, and it is available to families from Unique on request. This isn't super applicable to Gypsy, and it's super long and boring, but if you just want to learn about this chromosomal deletion, then stick around. Heart problem. It is uncertain whether having 1q21.1 microdeletion puts a baby at risk of being born with a heart problem. Some researchers have found a high incidence of heart problems, but others have not. In a study of the complex heart anomaly known as tetralogy of Fallot, 1% of affected children had a 1q21 deletion. Heart problems have not been identified among unique members. A gene known as GJA5 has been identified that may contribute to heart problems. The heart problems diagnosed in individual children may be single or multiple, as well as tetralogy of Fallot they include. I'm going to butcher these words, by the way. Patent ductus arteriosus, a channel known as the ductus arteriosus, is a normal shortcut in the circulation of the unborn baby. The channel usually closes naturally soon after birth. When it stays open, the lungs receive more blood than they should and the heart has to work too hard. It can be closed using minimally invasive surgery by inserting a coil via an artery in the thigh, or it can be clipped or tied shut. Atrial septal defects, ASDs. Holes in the muscle wall between the two filling parts of the heart. Some blood flows through the left to the right side, increasing the amount of blood flowing to the lungs. Treatment depends on the type of defect, whether it closes spontaneously and its size. Treatment can include medical management, taking medications to help the heart to work better, control of potential infection to the inner surfaces of the heart, and surgical repair with stitches or a special patch. Ventricle septal defects. Holes in the wall between the two pumping chambers of the heart or ventricles. This allows blood to flow from the left to right chamber, increasing the blood flow to the lungs. Treatment is determined individually. Small VSDs may close spontaneously. A larger VSD usually needs surgical repair to prevent lung problems that would develop from extra blood flow. Transposition of the great vessels. The main blood vessels, aorta and pulmonary artery leading from the heart, are reversed. A baby usually needs surgery very soon after birth. Truncus arteriosus. Instead of having separate blood vessels leading out of each side of the heart, a baby with truncus arteriosus has a single blood vessel leaving the heart that then branches into several vessels that go to the lungs and the body. This great vessel usually sits over both the ventricles, and the upper part of the wall between the two chambers is missing, resulting in a VSD. Early surgical repair is usually needed. Aortic valve abnormalities. The aortic valve regulates blood flow from the left ventricle into the aorta, the blood vessel leading to the body. The valve may be weakened or inefficient, causing blood to flow back to the left ventricle of the heart. The valve normally has 3 flaps or valves, but a bicuspid valve has only 2. In many cases, no treatment is needed, but a valve replacement may be needed. Aortic abnormalities. These include dilation of the aorta as it leaves the heart and coarctation, narrowing of the aorta. The narrowing forces the left side of the heart to pump harder to push blood through. Treatment is tailored to the individual, but if necessary, the narrowed section can be surgically removed or made larger. Another abnormality seen is interrupted aortic arch, where the aorta is blocked, leaving no direct way for red blood from the heart to reach body parts downstream from the blockage. It needs surgical repair as soon as possible. Loose joints or double jointedness. One study has pinpointed very loose joints or double jointedness in around a quarter of people with 1q21.1 microdeletion. The same study found that a similar number of people had a low muscle tone, hypotonia. In some people, the two conditions coincided. In others, they didn't. Among the unique group, low muscle tone was fairly common, but very lax joints were not seen. Early physiotherapy and regular exercises can improve hypotonia very much in some children. Children with very lax joints may need supports, splints, or special footwear while learning to walk. Increased risk of behavioral or mental health problems. Remember, in all of Gypsy's records, every time it said behavioral problems, they said no, and they talked about how good she was behaviorally. Children and adults with 1q21.1 microdeletion can be affectionate and sociable with no behavior problems, but as a group, they appear to be at risk for a range of behavior difficulties and mental health problems. We do not know yet whether these unusual behaviors and mental health problems are a result of the 1q21.1 microdeletion or a chance association. This is particularly true when a parent and child have a similar behavior pattern and both have the 1q21.1 microdeletion. So basically, they have no idea if this microdeletion even causes any behavioral disorders at all. The increased frequency of behavior difficulties suggests vulnerability in this area and means that children and adults should be monitored and families offered early support. The key concerns that have emerged so far are listed below. attention deficit with hyperactivity. This has only been observed in children with learning difficulties or developmental delay, which Gypsy did not exhibit either. Antisocial behavior. Antisocial behavior is not the same as antisocial personality disorder. Not the same thing. Seen in parent-child pairs and those whose deletion has not been inherited. Aggressiveness. This has been seen in children with learning disabilities. Autistic-like behavior. This has only been seen in children with learning disabilities. Anxiety. Seen in parent-child pairs and those whose deletion has not been inherited. Depression. This has been seen in parent-child pairs. Hallucinations. Schizophrenia is more common among people with 1q21.1 microdeletion than in the general population. It is possible that small head size may be somewhat involved, but the link is not yet clear. People with the 1q21 microdeletion and schizophrenia respond to neuroleptic drugs in the normal way. Unique families report marked mood swings in their children with fierce temper outbursts, screaming, aggressive, and stubborn refusal to conform. However, patterns vary and children and adults can also behave delightfully. Remember, Gypsy's doctors repeatedly noted how good her behavior was, and remember that the first published article about this microdeletion was how it correlated with schizophrenia, so it makes sense that we're talking about schizophrenia a lot here because it's like one of the only published articles. Their features: seizures. Seizures have been reported in 6 out of 55 people in the medical literature and in 3 out of 10 unique members. In the unique members, seizures started in the first year of life. In one case, they resolved by 2 years, but another child has had a type of seizure disorder known as Lennox-Gastaut syndrome with different types of seizures that are very difficult to control. An adult has absence, complex partial, and other unidentified seizure types that lead to long periods of being pale, sleepy, vacant, and out of it. None of those are the seizure disorder that Dee Dee was claiming she had. That doesn't even fit in her medical records, actually. Disturbances in the lens of the eye. Cataracts, clouding of the lens of the eye, have been seen in 5 out of 44 people with the 1q21.1 microdeletion, including 2 sisters, although their father, who also carries the deletion, had no cataracts. Cataracts have not been seen in unique members with the deletion. The type of cataract varies from being dense and diagnosed at birth to subtle cataracts only found after a specific search. Cataracts that cause no visual problems do not need treatment, but if the cataract affects vision, surgery to remove it is usually considered. A gene called GJA8 has been identified that may contribute to problems with the lens of the eye. In people with 1q21.1 microdeletion, other eyesight problems include long or short sight, a squint strabismus, Duane anomaly, restricted turning of eye associated with narrowing of the eye, abnormally small eyes, dislocated lenses, coloboma, a developmental defect, and double vision. Some problems can be corrected with glasses, although some children will not tolerate wearing them. Other problems, such as a squint, can generally be corrected with surgery. Unique has 1 member with this deletion who is registered as blind. Increased risk of other inborn anomalies. A wide range of other minor or more serious conditions identified at birth have been seen, but they don't seem to follow a particular pattern. They include missing ribs, extra fingers or toes, webbed or incurving toes, club feet, unusual brain structure, hydrocephalus, increased fluid in the brain, minor genital anomalies, birthmarks, tongue tie, hernia in the groin, cleft palate, lung malformation, small kidneys, renal hypoplasia, which can also have abnormal tissue, renal hypodysplasia, or absent kidneys, renal agenesis. Why are people with 1q21.1 microdeletions so different from each other? One reason for the difference is that the amount of missing chromosome material and number of missing genes is larger in some people, but people in the same family with the same size microdeletion can be very differently affected. We don't yet understand all the reasons for this, but one possible explanation is undiscovered genes. The 1q21 band of chromosome chromosome 1 has such a complex structure that its precise sequence has not been completely mapped. There are still 15 gaps in the map, and it's possible that genes exist in those gaps that contribute to the effects of the microdeletion. How did this happen? 1q21.1 microdeletions can occur out of the blue for no obvious reason, or they can be inherited from either the mother or the father. The only way to be certain is to check the chromosomes of both parents. The parents' chromosomes should be checked even if they are themselves completely healthy with no developmental problems at all. If one parent has the same microdeletion, it has been inherited. If both parents have normal chromosomes, the 1q21.1 microdeletion is a new occurrence. The genetic term for this is de novo. A new 1q21.1 microdeletion has been caused by a mistake that occurred when the parent sperm or egg cells were formed or in the very earliest days after fertilization. This is part of a natural process, and as a parent, there's nothing you can do to change or control it. At one point, all the chromosomes, including the 2 chromosome 1s, pair up and swap segments. To pair up precisely, each chromosome recognizes matching or near-matching DNA sequences on its partner chromosome. However, throughout the chromosomes, there are many DNA sequences that are so similar that it's thought the mispairing can occur. The 1q21 region has an extremely complex structure. 3/4 of it consists of at least 4 blocks of DNA that are more than 90% similar to each other. It is quite likely that this very similar blocks have caused a mismatch. When researchers examined the breakpoints in the chromosome in individual people, they found that they fell within these near-matching DNA sequences. In most people, the breakpoints occurred within parts of them that are virtually identical. Though no one has ever seen this happen, it is believed that when the exchange of genetic material known as crossing over occurs after mismatching, it is unequal, looping and cutting out a short length of the chromosome. What is certain is that as a parent, there is nothing you could have done to prevent this from happening. No environmental, dietary, workplace, or lifestyle factors are known to cause 1q21.1 microdeletions. There is nothing that either parent did before or during pregnancy that caused the microdeletion. If one person in a family with the 1q21.1 microdeletion is mildly affected, will others in the same family also be mildly affected? Not necessarily. There's a lot of variation between different members of the same family. We know that if one person is mildly affected, others may be more severely and obviously affected. Can it happen again? Where both parents have normal chromosomes, it is unlikely that another child will be born with 1q21.1 microdeletion or any other chromosome disorder. Very rarely, less than 1%, both parents have normal chromosomes by a blood test, but a few of their egg or sperm cells carry the microdeletion. This is called germline mosaicism, and it means that parents whose chromosomes appear normal when their blood is tested can have more than one child with the deletion. In families where the 1q21.1 microdeletion has been inherited from a parent, the possibility of having another child, either a girl or a boy, with the 1q21.1 microdeletion rises to 50% in each pregnancy. However, the effect of the microdeletion on the child's development, health, and behavior cannot be reliably predicted. Your genetic center should be able to offer counseling before you have another pregnancy. Remember, Gypsy owes us no information on this child or any future children, although she did tell us already that this child does not have this microdeletion. They already did the chromosome testing. Leave her and her future children alone. Do not ask her about them. She does not owe you any information. She has a right to privacy. Will my child with a 1q21.1 microdeletion have similarly affected children? Your child with a 1q21 microdeletion may well want to have children. We have not known about the condition for long enough to be certain if it affects fertility, but it is likely that fertility will be normal. In each pregnancy, someone with the deletion has a 50% risk of passing it on and a 50% chance of having a child without the deletion. Their ability to look after a child is very likely to be closely related to their own learning ability. Some genes in 1q21.1. The part of 1q21.1 that most people have lost is rich in genes. You can see the approximate position of 3 of these genes in the diagram on page 3. The HYD1N gene found on chromosome 16q22.2 has a part copy paralog that was inserted into 1q21.1 during evolution. The HYD1N gene on 1q21.1 is only active in the brain. In individuals in whom it is missing, head size is small, while individuals who have an extra copy of this gene have a large head. This strongly suggests that the HYD1N gene plays a role in determining head size. GJA5 is a gene that produces a protein known as connexin 40. It's expressed in the upper chambers, atria of the heart, and not having enough is associated with congenital heart problems. Some people who do not have this gene have normal hearts, though, and the reason for this is not fully understood. GJA8 is a gene that produces a protein known as connexin 50, whose job is to keep the lens in the eye transparent. The gene is expressed in the cornea of the eye and the retina of the brain. Some people who have lost GJA8 have cataracts, which can be very subtle or obvious. The GJA8 gene has also been associated with schizophrenia. That's the end of this 12-part series where we read this info packet from rarechromo.org. What are your questions?